I want to begin by asking a heretical question. For the evolutionary biologist, do we actually need to bother trying to find out what all the genes are doing? Do genes actually matter for evolution? That is, if I want to be able to predict how a trait is going to evolve through time, do I need to know, or is it helpful for me to know, what all of the individual molecular interactions are at the genetic level, at the biochemical level? That might sound like a crazy question, especially in the year of our Lord 2026, where we have dumped just billions and billions of dollars into molecular biology. The entire field is built on the back of the belief system that if we know what the genes are doing, then we can make powerful predictions about traits, right? That there's some sort of easy one-to-one connection between genes and the traits that individuals have. And it seems like that would be a really nice thing for the evolutionary biologist to know about. I want to play you a short clip by population geneticist Nick Barton. He gave this talk at a 2019 seminar, and this was actually for the 100th anniversary of R.A. Fisher's 1918 paper that we're going to talk about here shortly. Listen to what he says at the very, very end, and he does this in a sort of British tongue-in-cheek way, but I think the point is still made. Have a listen.
Where they argue that actually there may be an enormous number of genes, and even more provocatively argued that these genes affecting traits may not be concentrated in particular pathways. They may be spread over the genome over many, many different biological pathways. So then you ask, well, why bother finding the genes? Why not just live in statistical heaven? That's my argument. Okay, thank you.
So when I first heard that, I was a little befuddled myself. And I was like, what the heck does he mean by that? And he suggests that we live in a kind of statistical heaven. And I found that really curious. Over the past couple of years, I've been really thinking about this idea, about whether we can or whether we should live in a sort of statistical heaven when it comes to evolution. I was sort of pushed to finally give my thoughts on this, to finally develop a coherent way of thinking about this when two papers came out very, very recently. One of them, I'll show you, is from actually Nick Barton himself. This is from 2022 in PNAS, and it's titled The New Synthesis. And then a second one that came out just this February in 2026 titled Beyond Mendel, a call to revisit the genotype-phenotype map through new experimental paradigms. Both of these papers make basically the argument that Nick Barton was making, and the argument that I'm going to try to make to you in this video today. All of this is going to be given within the context of what I've decided to call the lost evolutionary synthesis. And that's only a little hyperbole. I hope to demonstrate to you that it's really not that hyperbolic, but it's a little hyperbole. Because what we're going to be talking about is basically an entire branch of evolutionary genetics that developed for over the past century almost completely out of the public eye. Very, very few people are aware of its existence and aware of just how powerful it is. Every single thing you eat has been shaped by this branch of evolutionary genetics. And what's particularly powerful about it, it's basically a genetic theory of evolution. And so I want to try to tell you that story, the story of the lost evolutionary synthesis. And in doing so, maybe just instill in your brain, plant a little seed, if you will, that we might actually not need to know much about genes at all to make really powerful predictions about evolution. Hello everyone, welcome back to the channel. I'm Zach Hancock. If you're new here, I'm an evolutionary biologist. I specialize in population genetics. On this channel, you typically find a mix of sort of evolutionary biology education. I do a little bit of pseudoscience debunking. I haven't done that in a long time because it's largely felt like a waste of my time recently. And a little bit of the sort of history and philosophy of science. And in this video, we're basically going to be doing the first and the third thing. I was sort of inspired to make this video even before I ever saw the lecture from Nick Barton that I showed you in the intro. And the story actually begins all the way back in 2021. And this is during the height of the COVID pandemic. And at this time, my grandfather passed away from COVID. And this is a really hard time for the family, as you might imagine. I'm sorry for the personal diversion, but I think it's important to understand the story. One of the things that you have to do anytime a relative, a loved one passes away is you, as a family, you get together and you have to go through their things, right? Because people leave behind a lot of stuff. The majority of that stuff is junk, but you do sort of have to sort through it, figure out who wants what, and you toss out the rest of it. And in doing so, I was going through his closet and I found this absolutely lovely book. It's called The Pigeon. You can see it's really old, it's falling apart, you know, the pages are all yellowed. It has a kind of smell to it, like a very old book smell. If you've gone into like a used bookstore, that really a pungent smell that old books have. That's what this book smells like. It's very nostalgia-inducing. It comes from a time in my grandfather's life when he raised fancy pigeons. His favorite breed were what are called Birmingham rollers. If you don't know anything about pigeons, Birmingham rollers are a group of tumbler pigeons who get their name from the fact that they have this kind of neurological tic, and it's a genetic neurological tic. Well, they'll fly up in the air and then they'll just like turn flips in the air. They'll do it in the air. They'll also do it if they're like standing on a perch. They'll just jump up and turn a flip, right? It's called rolling or tumbling. I don't have a ton of memories of it. I was pretty young when he was still raising pigeons, but I was really excited to find this book. This book is just entirely about pigeons, and it's written by this guy named Wendel Mitchell Levi. And Mitchell Levi is not a biologist. Did some like reading about him, and it turns out he was in the First World War, and he was in the like pigeon division of the Army Corps, which I didn't know existed. But sure enough, you can find these images of parachuters with like pigeons strapped to their chest, and apparently they're gonna like jump behind enemy lines, maybe do some recon or something, and then they send pigeons back to base. Levi talks about people doing it as far back as like Solomon, you know, sending pigeons to his armies in 3000 BC, right? It's like really crazy and cool stuff. So that's a little bit about Levi. He was apparently a pigeon fancier, right? But that's really all that he was. But he wrote this gigantic reference book on pigeons. It's really lovely, it's really charming and endearing. It has some very cute pictures and stuff in it. But the thing that really jumped out to me, there's a chapter called Genetics, Variation, and Inheritance. And this kind of got me excited because as I opened it, one of the first things that jumped out is that Levi, again, remember, he's not a biologist, he gives this history of inheritance that I find really fascinating. He talks through Aristotle, he talks about the Renaissance, he talks about Buffon, he talks about Jean-Baptiste de Lamarck, he talks about all these people. What he writes about Darwin is really fascinating. He says, Darwin's powers of observation were keen, and the mass of evidentiary facts collocated by him is astounding. He goes on to write that the origin of species was followed in 1868 by the publication of The Variation of Animals and Plants Under Domestication, and in 1871 by the publication of Descent of Man. Darwin's observations on pigeons in these works are numerous and are especially interesting to the pigeon lover. So one of the things that was really surprising to me in sort of doing some research about like Levi and about pigeon fancying and stuff like that is just how sort of obsessed pigeon breeders are with Darwin. If you go to the National Pigeon Association, which it's a website that exists, you can go to it and they refer to Charles Darwin as a scientific icon. The reason is because Darwin was a huge pigeon fancier. Darwin didn't actually get into pigeons until quite later on in life, but this is what he writes about them. He says, quote, "I have been led to study domestic pigeons with particular care because the evidence that all domestic races are descended from one known source is far clearer than with any other anciently domesticated animal. Furthermore, he writes, some domestic races of the rock pigeon differ fully as much from each other in their external characters as do the most distinct natural genera. He was very, very proud of his pigeons. There's a lot of letters of him writing to his friends, sort of trying to get them to come and look at his pigeons and see what breeds he's been crossing and stuff like that. His daughter Henrietta wrote about these pigeons like so. She said, I can still recall their different characteristics. A cross old fantail, when taking food from my hand, liked to take a good peck and hurt me if he could. The pouter pigeon was good-natured but not clever, and I remember a hen Jacobin which I considered rather feeble-minded. And so for Darwin, the pigeon fancying was sort of a really, really great jumping-off point for his own theory of natural selection. Because if humans can do this dramatic change in just a century or so, what can nature do having, the millions of years at its disposal? And that's basically what Darwin's argument was. Now, we all know that Darwin had one big problem: inheritance. We didn't know what the mechanism of inheritance was. And without a mechanism of inheritance, Darwin's theory of natural selection felt to many biologists that it kind of stood on shaky ground. And a big reason for this was because if inheritance is blending, that is to say, if children are just a blend of their parents, then what happens over time is the variation in populations gets lost. It gets blended away. And so there's nothing for selection to act upon. You can think about it like this. Imagine there's a population of organisms, and a new mutation arises that makes one of those individuals more fit than everyone else in that population. Well, that individual has to mate with someone in that population that does not have that mutation. And if blending inheritance is the way inheritance works, then their offspring only have half of the benefit of that original mutation because they've blended with the other parent that didn't have the benefit. And so now their offspring has only half the benefit. When they mate with someone, their offspring has only half of their half. And so you get this geometric decrease in how fit that organism is until eventually it just completely blends away. This was actually the argument made by an engineer named Fleming Jenkin, and it really, really troubled Darwin. It drove Darwin crazy. He spent a lot of time trying to figure out how to deal with this problem. So Darwin had an inheritance issue. Now what's really fascinating is that in Darwin's time, there were two people working on the problem of inheritance. Two big names that would ultimately form the sort of the heads of the two branches of the genetical hydra that I'm talking about in this video. One of which is Mendel. Mendel's genetics, Mendel's form of genetics would go on to really conquer the public consciousness. Like, that's the way we think about genetics to this day, as these discrete particles. There's a gene, that gene gives rise to a phenotype, can make crosses, and there's dominance and recessivity, and all of those Mendelian ideas, right, that you're all taught in high school biology. That dominates the way we think about genetics. And then there's another branch, and that other branch was led by a man named Francis Galton. And what's really fascinating about this is that both Galton and Mendel were born in 1822, the exact same year both men were born, and they were both working on this problem at exactly the same time. So we're going to start off, I'm going to give you the abridged version of the history of Mendelism, the history of Mendelian genetics, because you all already know it. But I think it'll be helpful to sort of recount the story as we're aware of it, and then I'm going to tell you the story of Galton. I'm going to tell you the story of the other branch of the study of inheritance that would become the field of quantitative genetics. One thing that's fascinating about Mendel that I really want to sort of start with is that Mendel was trained in mathematics. He was trained in statistics. One of his advisors at university was Christian Doppler, whose name is associated with the Doppler effect. Mendel actually taught high school physics for many years, so he had a really, really deep understanding of mathematics. And I think that is what prompted Mendel to approach inheritance in the way that he did. Because basically Mendel's idea was that inheritance is occurring not by blending, but there are these discrete particles, these indivisible particles that each parent has, and each parent has two of them, and they pass one of them at random to their offspring. Now, he didn't know anything about chromosomes, he didn't know anything about DNA, he just thought of them as particles. And you can kind of see the physics behind this, right? His sort of physics brain working here is because that's the language that a physicist would use in thinking about sort of fundamental particles or fundamental units of matter, right, as being particles. So that's how Mendel thought about it. So to explain Mendel's model, let's take a lesson from pigeon genetics. So in pigeons, there are several genes that follow the very simple Mendelian pattern. One is called the slipper gene. And this gene has two possible alleles, slipper or no slipper. What a slipper is in a pigeon is this sort of feathers over their foot. It looks kind of like they're wearing a little slipper. If you have two copies of it, then you have a full feathery foot, right? And your foot is completely gone. If you have one copy of it and one copy of the no slipper, so you're heterozygous, then you have kind of a half slipper. And then if you have two copies of the no slipper, then you have no slipper. So there's a few ideas here that are important to point out. One is the idea of diploidy. Diploidy means that you have two copies of the same gene. And that is really what we're saying is that you have two genomes. One of those genomes comes from your mom, one of those genomes comes from your dad. Very often you hear people say things like, you get half your genes from mom and half your genes from dad. That's not true. You get a complete set of genes from mom, a complete set of genes from dad, and then you just have two copies of every gene. That's what a diploid really is. So that's the basic idea of Mendelian genetics. And from that, you can do simple crosses and you can work out who is going to have what trait based on the known genotype of their parents. So in the slipper gene, you have three different possible traits. You have no slipper, half a slipper, complete slipper, depending on how many copies of the allele that you have. So pigeons also have what's called a crest gene. And this crest gene determines how much feathers they have sort of coming up over their face. It's a really sort of funny thing where it looks like their head is sitting in like a flower. And the no crest allele is dominant to the crest allele. So what that means is that if you have one copy of the no crest allele, then you don't have a crest. You have to have two copies of it to get that little neck thing. So imagine if you cross two individuals that are heterozygous for no crest. That means that they both have one copy of no crest, one copy of crest. You can very, very easily predict what the distribution of their offspring should be just based on what's called the binomial expansion. So if we let big C represent no crest and little c represent crest, then you can write this out, big C plus little c squared, which expanding it out is equal to big C squared plus 2 big C little c plus little c squared. And this is the famous 1-to-1 genotypic ratio that Mendel found. Phenotypically, it's a 3-to-1 ratio because on average, 75% of the offspring are going to have no crest, 25% will have the crest. And this is basic Mendelian genetics. Again, this is things that you learned in high school that you're all probably extremely familiar with. After Mendel published his work, it went basically unknown and unrecognized for a really long time. It was cited, it's not like it just completely disappeared into the void, but it wasn't cited for its discoveries about the mechanism of inheritance, right? That's the thing that was kind of lost. It was just like plant hybridization work is what cited him. It wasn't until the late 1890s and early 1900s that a group of German botanists basically rediscovered Mendel's work. And they did this by discovering Mendel's ratios first themselves, and then when they went to publish this stuff, they started doing like literature reviews because, you know, they were looking for like, who do we cite? You know, we're putting together this work on plant hybridization, and sure enough, they stumbled on these old German papers by Mendel and were like, holy crap, Mendel did all of this already and already knew these ratios. And so as this sort of history implies, most of the people that were doing the rediscovering and working on Mendelian genetics were doing so in labs, in gardens, right? They were crossing these highly inbred plants. I should have mentioned that one of the things that Mendel did so that he could work out the genotypic ratios is he took peas and he inbred them for many, many, many generations. He's basically wiping out all the variation that could possibly exist, And so that he had what's called a pure line. This is just a highly inbred line with no genetic diversity. And then he did the same thing with a different line of peas. So he created all these inbred lines of peas that only had one allele, right, and no variation. He did this so that then he could cross them and know that the exact expected distribution of the offspring, because he knew he was crossing purely inbred lines. And so all of the other geneticists basically followed suit with this, and this is how Mendelian genetics has worked for a really long time. In fact, we create these highly inbred lines, whether they're fruit flies, whether they're nematode worms, whether they're mice, whatever they may be, form the basis of how we understand Mendelian genetics. We want to get rid of all of that excess variation. That's how the Mendelian geneticists worked. Now, there was this guy named Wilhelm Johansen. He was one of the early Mendelian geneticists. And what he was working on was also pea plants, but he was looking at the inheritance of weight in pea plants. So he took his lines and he bred out all the variation, all the genetic variation, so they were just highly inbred lines. But what he found is, regardless of the fact that they had no genetic variation, they still displayed phenotypic variation. And whenever he plotted that, he found that the weight of peas in the offspring of these inbred lines was roughly normally distributed, so a continuous distribution, despite the fact there was no genetic variation. And what that told Johansen was two really important things. One is that there's a distinction between genotype and phenotype, and that genes lead to phenotypes, but that phenotypes can also be shaped by other outside things. These are maybe some environmental factors are also shaping phenotypes. The second thing is that because genes are discrete, right, they're these particles passed on from one generation to the next, the traits that matter, that are genetically controlled, are discrete traits. So think about like flower color, like are you purple or are you a white flower? Like those are two discrete different traits. Do you have a crest or no crest? Do you have a slipper or no slipper? These are traits that are discrete, and those are the things that Johansen believed correlated to genes. Whereas the continuous traits, the things like the weight of the pea or something like human height, those are continuous traits that are not easily one-to-one connected to a gene. He argued that while genes determine some parts of them, the majority of that was just environmental noise. And so studying continuous traits actually didn't matter that much. They were not important for either the study of genetics or the study of evolution, because evolution is fundamentally working on the inheritable component of the organism, not the environmental component. And so if continuous traits are just mostly being shaped by the environment, who cares about them from an evolutionary or genetic perspective? Johansen's impact on the field can't be overstated. It's super important to understand that the way that genetics developed was from this central idea that discrete variation is what matters at the end of the day. And it tells us something about why we still go out and try to find the gene that causes the trait. There's this one gene and there's this one trait, and there's this easy connection between them. And that goes all the way back to Mendel and goes all the way back to people like Wilhelm Johansen. And he also promoted the use of inbred lines, highly inbred lines, with removing all that variation so that you could study individual genes one at a time. Now that led, in the Mendelian case, of using extremely simple organisms in the lab. Because you can't use humans, you can't use complex organisms if you have to breed out all the variation. So that ends up being the critters that you use are C. elegans, E. coli, Drosophila, pea plants, right? Like these easy to rear organisms within the lab, and organisms that tend to be relatively simple themselves. So another key feature of Mendelian genetics is to uncover how genes work. You need variation, which means that sometimes you have to induce mutations. And the way that the early geneticists did this, people like Hermann Muller, people like Thomas Hunt Morgan, is they basically irradiated flies with X-rays, right? Like they just blasted them with X-ray radiation, and that led to these like big mutations, these big sort of monstrous mutations that they were then able to do crosses and uncover what gene had been mutated. But a key thing with that is that means that the mutation has to make an obvious impact on the organism. It has to be a visible phenotype. If a mutation occurs, but you can't see it, it doesn't seem to cause any kind of phenotypic effect, then you can't do anything with that, right? It ends up just being lost. It ends up flying under the radar of the Mendelian geneticist, right? Now, apart from just sort of predicting offspring phenotypic distributions, giving genotypic distributions, right? The Mendelian geneticists were also really interested in mechanisms. They wanted to know what is the actual sort of material of inheritance. And so Thomas Hunt Morgan in the famous fly room in Columbia, set out to try to answer this question. And he used Drosophila, he used fruit flies. And what they uncovered were that genes, these imaginary particles that Mendel sort of conjured up in his brain with a little bit of math, they realized that those genes are inherited on chromosomes, on discrete biological entities. That also gave people like Muller a target to mutate. So you blast a chromosome with X-rays, you can then mutate it, you can then map out what gene is where. That's what Alfred Sturtevant did within Thomas Hunt Morgan's lab. He did chromosome mapping to figure out where genes are, how close they are to one another. And then basically by the time we get into the late 1940s, 1950s, we now are working out the structure of the material of inheritance. We're now working out the structure of DNA, right? So people like Rosalind Franklin, Francis Crick, James Watson, all of these individuals end up working out that DNA is the material of inheritance, what its structure is. We then know where mutations are actually affecting, like what they're actually doing. They're changing nucleotides, they're rearranging chromosomes, those sorts of things. By the time you get into the 1960s and 70s, we've basically redefined the concept of a gene from the Mendelian idea as this discrete, indivisible particle, right, that's, again, just a mathematical construct to Mendel, to being a biological, biochemical thing, right, as a string of nucleotides that produce some functional product. Mendelian genetics becomes extremely focused on the molecular biology. So because of this, we funneled billions of dollars into molecular biology to try to uncover what these genes are doing. If you go from the Johansen view of phenotypes as being determined by genes, then it makes sense why you would want to do that. Because now we can think about traits in humans like Alzheimer's or diabetes or any of these sort of disease-causing traits that you might want to know the genes that underlie them. Because if you know the genes that underlie them, then you can like create medicine, targeted genetic medicine, gene therapy, to basically solve those problems. Fast forwarding now into the 1990s, you have the Human Genome Project. This was when Bill Clinton was president. Right after the first draft of the Human Genome Project was released, Bill Clinton stood in front of like a press conference and basically said that this is the most beautiful map we've ever created, that it's going to revolutionize human medicine, and that we were going to be able to basically solve almost every single human disease thanks to having this genome sequence. Francis Collins basically said the same thing. He said, and this is in the year 2000, he said in 15 to 20 years, we will see a complete revolution in the way in which medicine is administered in this country, and maybe around the world. What was going to cause that is the fact that we now knew the genes that were impacting the traits. If you know the gene that causes Alzheimer's, you just go in and you fix the gene. Boom. You have solved the problem. There was this really great article in the Los Angeles Times by this journalist named Laurie Garrett, and this was in 1996. And Garrett envisions a future in which a patient comes into the hospital and they're sort of catatonic and the doctors are trying to figure out what's wrong with them. And so they just like take out their wallet, pluck their DNA card out of their wallet, scan their DNA card, and they're like, oh, well, it looks like they're heterozygous for this mutation and they have this, this, and this mutation, so we know that they're going to need this and this and this treatment. That was in 1996 that they predicted that. And what's really kind of fascinating is in this article, she has a time frame. She has a future projection of when this futuristic society was going to be. It's 2021. And 2020, she's writing this in 96. By 2021, she thinks that we're all going to have DNA cards in our wallet, somewhere between our driver's license and our Kroger card. All of this, I want you to now juxtapose with the 2019 talk by Nick Barton that I opened this video with. This whole history that I've just given you has been all about genes, and all about the importance of genes, and all about the molecular biology of genes, and the billions of dollars we've put into it, with the idea that uncovering these genes are going to radically change human health. But any of us that were alive in the 2000s, and went to the hospital, at any point in time in the 2000s, and then has been in the hospital anytime in the 2020s, knows that the difference in treatment is not really that much. Think about the way we treat cancer today versus then. Think about the way we treat heart disease today versus then. It's the same. It's chemotherapy. It's a triple bypass. When was the last time you walked into a doctor's office and had your genome sequenced? Never. Most of us have never had our genome sequenced. Why? Why were these predictions in the 2000s, and these predictions, honestly, they go all the way back to Johansen, why did they turn out to be so catastrophically wrong? Why is it that today, despite having millions of human genomes completely sequenced, why do we still not know the cause of Alzheimer's disease? Why do we still not understand the cause of diabetes or heart disease or any of these things? We still don't know what genes are involved. We know that there's some genetic component, but it turns out when you actually look across the genome, when you scan all of the DNA letters in the human genome, it seems like most of them, basically any gene that is expressed in any tissues, has some correlation with basically every complex human trait. Whether it's human height, whether it's diabetes risk, whether it's schizophrenia, whatever it might be. When you look across the human genome, you find hits for everything, all over the place. That's a far cry from Johansen's idea that you just remove all the variation and that lets you find the one gene that's causing the trait. Because it turns out all that variation is all contributing to the trait. There's this great paper that came out in 2017, and they propose what's called the omnigenic model. And the omnigenic model basically works like this. It proposes that there is not one gene or two genes, or not even polygenics, not even like 20 genes. It's that every single gene that is at any point in time expressed is contributing to the variance in a trait. And their idea is that you've got a couple of core genes, and then all of those core genes are being interacted with by a whole dense network of genes. This large, infinitely complex network where every little gene and every little regulatory pathway is impacting every other one. Importantly, it's doing so in extremely tiny ways. So tiny, in fact, that when you look across the genome and you try to find correlations between those individual alleles and the trait itself and the variation in the trait itself, you don't get enough statistical power to say what gene's causing it. Right? Because the cause is so little, it's so slight. In short, it seems like the way most complex traits are not determined by a couple of genes, but they seem to be determined by every gene, every single gene that you have, and the contribution of each one of those genes is extremely slight. So slight, in fact, that it's virtually undetectable. And that's what Nick Barton was ultimately referring to when he asked the question, why bother finding the genes? Because if it turns out that that's true, that the genes are contributing so little to the variation in the trait, then finding out what any one of them does tells you almost nothing about that trait, because its contribution is tiny, and it's probably contributing to it from so far away in this highly dense network that drawing the connection back to the trait is basically impossible. It's so far away. It's meaningless at this point. So what do you do then? That finally brings us back to Barton's big question, and brings us back to Barton's potential solution. Why don't we just live in a kind of statistical heaven? What would that statistical heaven look like? So now I've given you the sort of story to Mendel, from Mendel all the way up to basically the modern day, to at least the Human Genome Project, and to the omnigenic model, to Pritchard's omnigenic model. What I'd like to do now is rewind us back, all the way back to 1822, when these two men were born, and tell you the other story. The story of the genetics that you haven't heard about. Genetics of statistical heavens, and using the techniques of mathematics and statistics instead of relying on the techniques of molecular biology. And hopefully in doing so, I can promote a view that if we can re-merge these two fields, if we can pull these two fields back together, then we can actually start to understand traits. I know I've been maybe a little bit doomer in this video so far, but I think the final synthesis, finally bringing these two things back together, will create the most powerful, unified theory of evolution that could possibly exist if we could get these two fields to communicate with each other once more. So I want to turn back the hands of time and let's talk about Francis Galton. There's a kind of funny irony about the history of Galton and Mendel and inheritance, in that in Mendel's day, virtually no one knew who Mendel was, and yet today, everyone knows who Mendel is. Everyone's familiar with the name Gregor Mendel. And yet, the exact opposite was true in Galton's day. Everyone knew who Francis Galton was in the 1860s. He was world famous. He was a renowned biologist. He was the half-cousin of Charles Darwin, after all. He actually wrote a highly influential book called The Art of Travel. This is a really wild read. You can find it online for free. It is just riddled with racism and extremely offensive things. It's basically the tale of a British explorer that goes to Africa. It includes things like how to properly holster your gun on a donkey, how to take a prisoner in case you are in a situation in which you need to, I don't know, take a prisoner. Apparently if you put your knife in your mouth, that's a really spooky thing to do. I don't know, it's full of really, really wild things. But it made him famous, right? His exploration through Africa, his writings about it made him famous. However, today, very few people know the name Francis Galton, and most of the people that do know the name Francis Galton know him as associated with eugenics. So if you didn't know, Francis Galton coined the term eugenics. He's considered the father of eugenics, and just in case you're unaware of what that means, you've been living under a rock for the past century, eugenics is the idea that we can direct our own evolution. And in particular, we can direct it in such a way as to improve the human stock, right, is the way he would have worded it. We can make people smarter, we can make people stronger, etc., by basically selectively breeding humans. Obviously atrocious and horrendous idea that led to some of the greatest atrocities in human history. So again, most people that know who Francis Galton is know him from sort of that angle. What we're going to talk about is what he's less known for today by, sort of the general public, but this is easily his most important lasting legacy in sort of the history of science. So this historian of science once referred to Galton as the most important Victorian scientist that you've never heard of. So let's try to get ourselves in the mind of Galton and how his sort of theory of inheritance worked. And so the way to think about this is, imagine you knew nothing about genes. Right? Let's take the whole Mendelian thing out of the way. Just wipe it out of your mind, roll back the clock to 1860, and pretend Mendelian genetics didn't exist. But what you wanted to do is you wanted to be able to make a prediction of what traits a child will have, or what traits a relative is going to have, given that you know the traits of their parents or some other relative. That is to say, you want to be able to estimate the correlation or the resemblances between relatives based on the degree of relationship. The language that we're using here, that we've kind of developed here, is clearly statistical language, right? We're looking for correlations between traits. And in this way, you don't necessarily need to know what the underlying cause of that correlation is. That is, you don't need to know what the material of inheritance is being passed from parent to offspring. All you need to be able to do is predict what it will be based on the degree of relatedness. So that is what Galton, or the way in which Galton sort of approached this problem. Another thing that Galton was really interested in, and this will play a really big role in sort of the history of these two fields here in a moment, is that Galton was interested in the observation that species seem relatively stable. What do I mean by that? So humans have been looking at organisms for a very, very long time, and we've been able to give names to species and recognize species for thousands of years, in fact, going all the way back to Aristotle. And while there's variation, and every single generation we see children look a little bit different from their parents, over time, you actually see lots of morphological stability. Now, again, that doesn't mean that change doesn't happen. It's just really, really slow, and species as a sort of a concept appear very stable. And so why is that? particularly given the observation that children can look quite a bit different from their parents. And so what you would expect, what Galton sort of logicked, is that, well, if selection is always acting, and children always look different from their parents, shouldn't any of the defining characteristics of a species disappear extremely rapidly? And why does it seem to be that species are relatively stable entities that only seem to change after really, really long periods of time? What's ensuring that stability? So those are the two sort of pillars of his approach. Why are species stable, and can I predict what traits a relative is going to have, given that I know the traits of some other relative of known degree of relationship? Okay, so that's his approach. So what did he do? Now what the Mendelians did is they crossed a bunch of things in the lab, right? They created these inbred lines, they crossed them in the lab, and they tried to work out the mechanistic rules of inheritance. Galton didn't think that way. And probably a key reason is because Galton was really interested in human evolution. As a eugenicist, he wanted to know what shapes inheritance patterns in humans. In particular, he was sort of trying to buck this idea at the time that humans are born blank slates, right? That we don't have any sort of inherent bias in the way we're going to act, the interests that we're going to have, those sorts of things. We don't have those kinds of inherent biases. And so if that's true, right, if we're born blank slates and there's no sort of genetic underpinning, then you can't really have a eugenic program. You can't selectively breed IQ in humans, right, if IQ is just a product of education. And so Galton set out basically to prove, in his mind, that these sorts of cultural traits were also inherited just like physical traits. So that was sort of his approach. And so because of that, he needed to look at the variation in populations. He couldn't just inbreed out all the variation, because it was the variation that mattered to him. So what did he do? What Galton did is he collected a ton of data for a bunch of different human traits. So for height, for grip strength, for breathing capacity, everything you can imagine. And what he found when you plot all of those traits is they form what is called a normal distribution. And a normal distribution looks like this. It's the sort of classic bell curve, right? Where here in the middle is what we call the mean. This is the mean of that distribution. And then around that mean, there is what is called the standard deviation, which is often symbolized as sigma. Importantly for this discussion, we'll also introduce a term that is sigma squared, and that is known as the variance. The reason you might want to know variances instead of just standard deviations is because if you try to sum up standard deviations, they also have negative values, because they can be less than the mean. And so if you sum them all up, you can end up like canceling terms out. Variance is a particularly useful way of looking at this, because since they're squared, they're in effect the absolute value, and you can sum them up without losing value. So that's what makes variances so useful for this. But anyway, so he found that most traits in the human population follow this sort of normal distributions. But what happened whenever he compared the values of one relative against the values of another relative? So I'm going to draw you out sort of a hypothetical. So this is going to be our graph. On the x-axis, we'll call this parents, and this might be something like height, like human height. And over here is offspring. So this is their children, right? And now what you would expect if children were just sort of clones of their parents, right? If every single aspect of a child's trait is inherited directly from their parents, such that they are like Athena from Zeus's brow, right? Like literal clones, then what you would expect is that the traits, I'm going to draw this as best as I can, but will be perfectly correlated between these two. And you could draw a line through them, and they would be just a perfect one-to-one correlation between parents and offspring. That's what you expect if they are clonal. However, what Galton found was that it didn't look like that at all. Instead, what he found were points that looked something like this. Points that looked something like this. And then here, we'll draw, this is the population mean, right? So this is the average of the population. This line is the hypothetical one-to-one fit. That's the hypothetical one-to-one fit. And what he found was the relationship between parents and offspring actually looked something like this. So it was different from the one-to-one fit, and in fact, it was closer to the average. That is to say, that children from, let's say this is height, children from really, really short parents tended on average to be taller than their parents. Okay, so children were taller than their parents at the bottom end, but then parents that were really, really tall tended to have children that were shorter than them. Right? And in both directions, they tended to be closer to the population average than what their parents were. Okay? And this concept is what is known as regression to the mean. Regression to the mean. Now, this is an extremely important discovery. It literally is what gives this analysis its name. The reason that we call linear regressions regressions is because of Galton. It's because of this. Galton referred to this as regression to mediocrity, but we call it regression to the mean. And so what this line here represents, what the slope of this line represents, is the degree of regression back to the average. We're just going to call this, for now, we'll call this R. And R stands for the regression coefficient. Okay? So again, it's a measure of the slope, but what that slope is actually indicating is how much more similar the y-axis is to the population average than the x-axis is. That's why this is called a regression coefficient and why we call linear regressions regressions. So what's the sort of implication for this? So remember, Galton is interested, one, in predicting the values of offspring, given he knows the values of parents. It could also be any kind of relative, but he wants to be able to predict one or the other. So the key thing here of making that prediction is being able to estimate what the regression coefficient is. Because the regression coefficient is the correlation. It's the correlation between these two things. So if you know an x value, you can predict a y value if you know what the regression coefficient is. And the way that he estimated this is what's called a linear least squares. I won't go into the calculus of how you actually estimate it, but there's a key thing that you need to know about linear least squares. And what linear least squares finds is that the slope ends up being equal to the covariance between x and y divided by the variance in x. And what linear least squares is in effect doing is it's taking a line, it's drawing a line through the points such that it minimizes the distance between those points and the line itself. So you set the error equal to 0 and then you solve. And what you end up getting is this slope will minimize the distance between these lines, okay, or distance between these points. That's the linear least squares approach. This approach was invented by Friedrich Gauss, right, in like the 1600s or something. So it's really, really old. We've been doing it for a long time. That's what the slope is going to be equal to, and this is going to be really important. So a covariance is a measure of the degree in which two values travel together. So for example, if I increase the variation in x, does the variation in y also increase? If so, there's a positive covariance between x and y. If, however, I increase the variation in x and that decreases the variation in y, then there's a negative covariance between them. And if I increase one and nothing seems to happen to the other one, then there's no covariance between them. Okay? That's what covariance represents. It's just a measure of how the variances in x and y travel together. All right? That's what the regression coefficient is going to be equal to. So one thing that Galton found that's really important is that using this, he is able to begin to predict the traits of offspring, given that he has the traits of the parents. And he's done so without any genes. Right? There's no genes involved in this. It's just correlations. Okay? But, again, Galton wanted to be able to explain why this happens. Why does it seem that offspring are more like the population average than their parents are? What can explain this? And so Galton put together a model of inheritance, okay? And his model of inheritance is going to surprise you in how close it is to Mendelian inheritance. And in fact, I'll show you later, he was almost right. He was really, really close to getting this right. So I'm going to walk you through what his model is, and what you're going to see is that it's a statistical model that just gets the biological details wrong. So Galton's model is called the law of ancestral heredity. The law of ancestral heredity. And again, what he's trying to predict is the deviation from the population average of a child, given that he knows the deviations from the parents. So we can write this out as what's called a linear model. So we'll write D naught is equal to the regression coefficient of parents, which is D1, the regression coefficient multiplied by the deviation in the mean of parents, plus the regression coefficient multiplied by the deviation in mean of grandparents, plus the regression coefficient times the deviation in the mean of great-grandparents, all the way out to the last sort of individual. Plus what he calls this little E term. And this E term is an error term. But in this model, you can kind of think about it as some environmental variation, right? So something that's not being inherited, but that's contributing to differences in the traits, right? It's contributing to some error in your ability to estimate this child's trait. What this is called is a multiple linear regression. That's what this is in statistics, because you've got regressions, regressions, regressions. You're summing up all of the different regressions, right? So now we need to know, what does this R represent? What is his actual model? So he predicts that what the R is, and we'll write it as R sub I, so the regression coefficient for some resemblance, is equal to P, I'll explain what that is in a moment, multiplied by 1/2 raised to the I. Now what does this mean? What this P value here represents is the probability that a particle that's inherited from some ancestor is turned on or turned off. So the idea that he had was a kind of particulate inheritance. He believed that parents are passing these particles to their offspring, but if that particle is turned on, then they express the trait that their parent had. If it's turned off, then they don't express the trait that their parent had. And so what it represents is the probability that you are expressing the same particle that an ancestor expressed. That's what it represents. Because you can imagine, if your parent doesn't express the particle, but an ancestor did, and so you end up expressing it when your parent didn't, what that means is you are going to look more like your ancestor than you are to your parent. And what that will do over time, if you sum that all up, that ends up equaling the population average. And so the offspring will consistently start to look more and more like the population average relative to what their parent looked. That's what this represents. And that was the way he tried to explain why regression to the mean occurs. So this is the probability that a particle that you have is on the same state, off or on, relative to the ancestor I. And that ancestor I might be the parent, might be the grandparent, might be great-grandparent, whichever. And then you just multiply 1/2 raised to the degree of relatedness, which is I. So if this is your parent, this is equal to 1. And it is half because you get half of your particles from a parent, right? Again, this is almost Mendelian inheritance. It's really, really close. You get half of your particles from your parent, but you get 1/4 of your particles from your grandparent, right? And so with each degree of relatedness, you get fewer and fewer and fewer particles from them that could be correlated here. And so this regression coefficient, with further and further terms, will get smaller, smaller, and smaller, because you will be less correlated with them over time than you are with your parent. This is a pretty incredible model, and it's mathematically correct. And I'll show you why. I'll show you how it's mathematically correct. There's one thing that's wrong with it, though, right? And I'm sure everybody watching knows what's wrong with it, is that this term doesn't mean anything. Right? This is not a term that means anything in the Mendelian sense. And so the biological interpretation of this model is incorrect, even though the math is correct. And this ended up taking Galton down this really wild sort of side road as he tried to calculate what this value is. Because in Galton's model, this value should be a constant. It should be a constant because it's a property of the mechanism of inheritance itself. It's a property of the genetic system, and so it should be constant for all traits. Every single trait should have the same p-value. But when he estimated the regressions for a bunch of different traits, he found that p didn't equal that. Because you can basically just algebraically rearrange this. If you can calculate the regression coefficient empirically, as I just showed you, you don't know what p is, but you can calculate this, you just algebraically rearrange it, and that gives you the p-value. Well, p ends up being equal to a whole bunch of stuff. He calculated 1/3, 1/5, 2/3, it was just like a bunch of different values, and he couldn't seem to figure out why. Right? That didn't seem to correspond with what his model of inheritance was, that p should be these particles turning off and on. But that's the law of ancestral heredity, and this law ends up being the flashpoint of the debate between one sort of branch of Galton's descendants and the Mendelian geneticists. Now, the last thing I should tell you about Galton is that Galton believed that his law of ancestral heredity and the idea of regression to the mean fundamentally challenged the Darwinian view of evolution as the most important kind of variation in evolution is slight and continuous. That's what Darwin believed, right? It was very, very little differences between individuals is what's going to drive evolution. But remember what Johansen said. Johansen argued that it's actually the discrete variation that matters because it's the discrete variation that underlies Mendelian genes, right? And so remember he had that curve of weights and p's, and despite the fact that there was no genetic variation, this is why he separated genotype and phenotype. Galton is basically agreeing with this. Galton is saying that because of regression to the mean, children are going to just look more like the average, and so selection is going to be ineffective against them. It's not going to be able to actually drive change. And so what you need big leaps. You need these big mutational, discrete leaps to transform the population from one generation to the next. You can't just have little slight variations. Now, what's really fascinating about that is Galton kind of serves as the forefather of these two views. Right? He serves as the forefather of Mendelian genetics as well as the statistical approach to evolution, which is quantitative genetics. And the reason for that is because he believed that inheritance, or that the most important kind of variation was discrete, but he developed the tools of quantitative genetics, right? And so he becomes a sort of forefather here. I now need to point out to you why Galton was wrong. And in fact, his star protege, a guy named Karl Pearson, recognized that Galton was wrong and made an argument as to how selection is actually extremely effective on continuous variation. And that's what I'm going to sort of walk you through next. All right, so who is Karl Pearson? Karl Pearson is another one of those uber controversial dudes in biology. Like, you want to like Karl Pearson. Because he developed this really elegant mathematical theory of selection based on a statistical approach, based on an objective quantitative approach. You want to like him because of that. He also developed a good half of modern statistics. R.A. Fisher did the other half, right? So he's really, really important in modern statistics. He was also a vocal advocate of women in science, especially in statistics, and the field that he represents is what's called biometric. He had a whole group of women that studied under him, and he regularly published with them. And this is a big thing. Like, in his day, women were doing a lot of the calculations for these studies, were not being credited in the paper itself. But that was not the case with Pearson. Pearson would often publish with them, including with people like Alice Lee, who was a really important statistician that worked with Pearson. There was a belief at the time that women had lower IQs than men because men had larger skulls and therefore larger brains. Right? So it was like a physical thing, that men have bigger brains than women, so men are smarter than women. And what Alice Lee showed is that she collected a bunch of data, right, measured skull sizes, and then she looked for correlations between skull size and IQ and found that none existed. So there was no correlation there. And so she, in effect, disproved that sort of misogynistic idea that men were smarter than women because they had bigger brains. And Pearson fought for her. This was her dissertation work, and he actually fought against Francis Galton, who didn't believe that could possibly be true, right? So Pearson supported her and supported lots of women in science. So for those reasons, you want to like Pearson. But then there's all this other stuff that you just can't like Pearson for, right? In particular, he was a eugenicist, just like Francis Galton. He even praised Hitler in the 1930s. Now, this is before the Holocaust, but like, he even stated something along the lines of he was excited at Hitler's fervor to rejuvenate the German people, right? Like that kind of thing, that's weird. He was anti-Semitic, like really, really disgustingly anti-Semitic, so Pearson is, you can't actually like Pearson for these reasons. So that's the kind of guy we're fixing to talk about. That's Karl Pearson. For our purposes, what matters for sort of this discussion, now that we've sort of given his background, Pearson is disagreeing with Galton over whether selection can act effectively on continuous traits. And he argues that yes, it absolutely can, and he shows how it can. So what we're going to do is we're going to go back to pigeons again. We're going to talk about the inheritance of squab weight. So if you don't know what a squab is a young pigeon that has not yet reached maturity and been able to fly. Apparently, in large parts of the world, squabs are eaten. They're like sold for food. And in fact, Wendel Levi actually opened one of the first squab production factories here in the United States, in actually South Carolina, not very far from me, in 1923. So that's the trait that we're going to use. We're going to think about the inheritance of weight of squabs, okay? So what Pearson showed is if we start off with this sort of graphical approach, and we will do parent weight on the x-axis, and then we will do offspring weight on the y-axis. And then we've got some points. This is parents and offspring points like this. Then we have some average. This is going to be our average here. We will write that as mu. That's our average. I'll draw a little line like this so we can see it well. And let's say that the original average of the population is 300 grams. 300 grams is the average of the parent population. Now we then need to calculate the regression. So we do our little line, we use our linear least squares, and we calculate, let's say the regression between parents and offspring is 0.5, so 50% correlation between parents and their offspring. So Pearson now asks us to imagine that we select some new average, okay? From that parental population, we allow a new average to breed. And we will draw that new average as right here, and we will write that as mu sub s, okay? So this is where we are selecting our new average. And let's say mu sub s is equal to 400 grams, okay? So now what we're going to do is we're going to draw that also here on the offspring. So this is mu. And then what we need to draw is where mu sub s is. So that's where we selected from here. We're just writing it up here. And so what we do is we go up the offspring weight over to mu s, and then we work our way over until we intersect with this mu sub s right here. That's the intersection point. And then we go down to the regression here. So notice that now the new average is going to be here. This is mu naught. This is now going to be the expected new average of offspring given the correlation between parents and offspring after we have done selection. So we can calculate this. So this is me showing it in a sort of graphical sense, but we can actually do the calculation pretty simple here, right? So what we need is what we're going to call S. This is the selection differential, and this is going to be equal to mu sub s, the average we selected for, minus the original average, which is mu. That's 400 minus 300, so that is equal to 100 grams. Okay? That is the selection differential. That's what this difference is. Now we want to know the response to selection. That's going to be calculating mu naught. What is that new average going to be? So we'll call the response to selection R, and it's going to be equal to the selection differential S multiplied by the regression coefficient. How similar are parents and offspring? So S times little r. What that is then going to give us, we know that S is 100, so that's 100 times 0.5, and so what we expect the response of selection to be is 50 grams. 50 grams is how much we can increase the average in one generation of selection, given that this is our selection differential, how much we selected for, and this is the correlation between parents. So what is mu naught? Mu naught is then equal to mu plus the response to selection, and so what we end up with is a new average of 350 grams. Okay? That is what mu naught is going to be equal to. Now notice something important here. Mu naught is not equal to what we selected, right? We haven't actually increased the average by the total amount that we picked from. And the reason for this is because children are not clones of their parents. They are only 50% similar to their parents, which means that we can only increase the average by 50% of what we selected for. And this teaches us a very important lesson, is that the more and more similar, the greater this regression coefficient is between parents and offspring, then the stronger, the more efficient selection can be, right? If this, and at the highest point, that it's 1, right? If they were exactly equal to each other, then this selection differential, would end up just being equal to the response to selection, right? But always, parents are some value less than 1. This is the idea of regression from sort of Galton's work, right? But what this also demonstrates is how wrong Galton was. That actually selection works just fine and can be quite efficient even when populations are regressing to the average. So this was Karl Pearson's sort of response to Galton, is that Galton and all the Mendelian geneticists who are arguing that you needed to have discrete variation for evolution to work were just wrong. They didn't seem to understand that you are changing the average. You are picking a different average. And that will overcome regression, given that the regression is not 0, right? If there's literally no correlation between parents and offspring, there can be no selection. But so long as there's correlation between parents and offspring, you can get a response to selection. I hope it is clear here that we have made no appeal to genes. There's no genes in any of this. And yet, we have made a really, really powerful argument about how selection is expected to act. And so one of the most remarkable things about Pearson's model is you can apply it to anything, irrespective of whether Mendelian genes are what is underlying that trait. It's just based on the correlation between parents and offspring, or between relatives. It's completely agnostic to the actual mode of inheritance. And this was a lot of the argument in the early 1900s between the biometricians and the Mendelians, is the Mendelians were like, you guys are not even working with anything biological. You're just looking at statistics, you're just looking at correlations. We want to know what's actually happening. Pearson argued that what's actually happening doesn't matter. He famously wrote, the answers to these problems, the solutions to these problems are in the first part statistical, and the second part statistical, and only in the third part biological. However, Pearson's model is not inconsistent with Mendelian genetics. Now, the Mendelian geneticists believed that it was, but there was a guy who emerged during this time, and he was actually a teenager when a lot of the most fervent debates were happening, but by 1918, he published a paper that showed the unification of the sort of Galtonian statistics and Mendelian genetics. That Galtonian statistics was completely consistent with Mendel's laws. What's really fascinating about this paper is that that sounds like an enormous accomplishment, right? To be able to show that those things are consistent with each other. And yet it was initially rejected. And it was rejected by 2 reviewers, one of which was Reginald Punnett. You should recognize this name from the Punnett Square fame. Reginald Punnett was an ardent Mendelian geneticist. He wrote one of the first textbooks on Mendelian genetics, so very, very strong on the Mendelian side. He rejected the paper because he referred to this person's math as being akin to weightless elephants on frictionless surfaces, right? He rejected the sort of mathematical modeling. The other reviewer that rejected it was Karl Pearson himself. And Karl Pearson rejected it because it was too Mendelian. it relied too much on Mendelian genes, and Pearson was a fervent critic of Mendelian genetics. So these two individuals, the geneticist, the biometrician, could not see that this paper had actually unified their fields and shown how they were both correct. Both of their fields actually worked together. And that all the animosity between them, that in thinking that it had to be one or it had to be the other, was just wrong. There was no reason to think that way. Of course, I've buried the lead. This person is R.A. Fisher. And the paper that he wrote in 1918 is called The Correlation Between Relatives on the Supposition of Mendelian Inheritance. And so the task before me is to try to demonstrate to you, as simple as I can, Fisher's model and how Fisher was able to unify these two fields. I'm going to try to do it with as little math as possible, but it is a mathematical model, so we'll have to sort of talk through some of the details. But what we're going to see at the end is that Mendelian genetics are actually entirely consistent with all of the mathematics that I just did from Pearson's model and from Galton's model. And they actually explain those things in a sort of mechanistic way. So how do we do this? One of the things that I need to warn you about Fisher's model is that Fisher's model is a little like magic. It's like magic in the sense that there's nothing about it that's real. We are going to travel into a land of make-believe, okay? We're going to assume a bunch of things that we can never know. You can literally never know the things that we're going to assume to derive this math. But at the end of it, and this is really the magic of this model, is we're going to come out of the other side, out of this little land of make-believe, we're going to come out of the other side with a practical model that has been one of the most powerful models in all of biology. In fact, this model has impacted your life so profoundly that you don't even know it, you don't even recognize it. But every single thing that you eat, right, all the crops that we consume, all the livestock that we consume, were bred and raised with this model as the guiding force of them, right? So I hope that at least demonstrates their power, and I'll give you some specific examples once we sort of work through this math. So first, let's imagine we constructed a model that explains the traits that an individual has, okay? And we're going to call this individual, we're going to say P sub I, so this is individual I, and P is going to be their phenotype, P for phenotype. And we're going to assume that their phenotype is composed by the sum of a bunch of Mendelian genes, all their genes, right? So maybe they have a genotype big A, little a, plus genotype big B, plus genotype little c, plus genotype big D, plus all the way out to some error term. Okay. And so this is the genotypic effect of gene A, B, C, D, etc. All of their genes. Every single gene that might be impacting that trait, you imagine summing them all up, and then at the end of the day, you've got some error term, and this represents the environmental variation, right? Something that's biasing your prediction, that's not a component of your genes, okay? That's the simple model. Now what we want to do is we want to be able to predict the traits in offspring, right? But a key problem with this model is that these are genotypic effects. That is to say, these are effects of genotypes, but what offspring get from their parents are not genotypes. They get alleles, right? You pass an allele on to your offspring. You don't pass on your genotype. Your genotype are two alleles. You're only passing on one. So we've got that sort of issue here. And so what Fisher asks us to imagine is we could break up the associations between genotypes and alleles. We could basically partition out that variation. And this is how he asks us to imagine doing that. Is imagine we could take any individual genotype and we could draw sort of a regression like this. All right? And we have here, there are two possible alleles, big A and little a. On the x-axis, we're going to draw how many copies of that allele do you have. So you either have 0 copies, you have 1 copy, or you have 2 copies of, we'll say, the big A allele. Now you're going to do the same thing with a little a allele, but for now we'll just do it with the big A. And then this is going to be the effect of those alleles. Okay? So let's draw out some effects that we can make believe. So let's say this one leads to this effect, this one leads to this effect, and then if you have 2 alleles, you have, let's say, this effect. Now what you're going to notice about the way that I've drawn this is that it's almost dominant. Big A is almost dominant. Because if you have one copy of the big A, it's very similar in its effect to if you have two copies of the big A. Right? So there's some dominance in this system. One of the magical things you should notice here is that we're assuming we can estimate the effects of each and every genotype. And we're assuming we're doing this for every single gene you have. Okay? And in fact, in Fisher's model, he's assuming an infinite number of genes. And that all of these effects are, in effect, infinitely small. Okay? We'll talk about the implications of that here shortly. But we're pretending we could do this. So those are the effects. Now the second thing that we need to bear in mind is that what we care about is the average effect of A within the population. On average, if we were to draw individuals from the population, right, how much is A going to be impacting their phenotype? Okay? And that's a way of measuring the allelic effect. That's a way of breaking apart the genotypic effect from the allelic effect, is thinking about, on average, what is A doing to your trait? Okay? But that means that it's not just the effects of A, it's also how common is A in the population. If A is very, very common, then picking an individual at random, more than likely they're going to have A, the big A, right? So we will sort of weight this. Let's say A is extremely common, so we'll draw this circle bigger, and this just means that it's the frequency. Okay, so the frequency of A is large. So we'll draw this one a little bit bigger, and then this one we'll keep small. Okay, and so the frequency is going to weight our regression. All right, and so now what we want to do is we want to draw that regression. So since these are going to weight it, we're going to draw it a little like this. And that's going to be the regression between these. Again, this is a regression via linear least squares, just like we did in Galton's model. So these are the actual genotypic effects, but then we have what is going to be called the average effect of each one of those alleles. So if you have 0 alleles of the A, the average effect he is defining as what falls along that regression line. Okay? This is the average effect of, which we'll call alpha 1, 1. Alpha 1, 1 is the average effect of having no big A alleles. That's the average effect. And then the distance between the average effect and the genotypic effect is what we're going to call the dominance deviation. This is the deviation between the genotype's effect, right, having big A, little a, big A, little a, and having no copies of the A allele, right? So we're breaking up that contribution. We'll do the same thing here. This is the dominance deviation of 1, 2, and this is also the additive effect, the average effect of 1, 2, and these are almost on the line, so the average effect is very, very similar. So that is the way in which we're now partitioning up these two things. So in effect, we've broken up the genotypics effect, which is the dominance deviations, from the average effects, which are the alpha values that fall along that regression line. Okay, so how do we want to write that? Well, we can just simply write the average effect of the big A allele is then equal to alpha 1 plus the dominance deviation between alpha 1 and alpha 2. And alpha 1 and alpha 2 are measures of the average effects of big A and little a. Okay, so in this way, what we have done is we have now broken up the effects of the allele, which is alpha, that's the average effect of that allele, which is estimated by the regression, from the dominance effects of that allele, which you can think of as the genotypic effects of that allele, which is the distance between the average effect and the genotypic effect itself. If you're familiar with regressions, you'll notice that this is also called the residuals. Whenever you're measuring this for something else, the distance between the points and the line itself is called the residual variance. Okay, so that's what that represents. All right, so now imagine that we do this, right? We calculate the regression coefficient for each and every genotype and every single allele, and we break up each and every one of them. So let's write this out. So let's say the genotypic contribution of individual I is then equal to alpha 1 plus, we'll say alpha 1 big A plus alpha 2 little a plus the dominance deviation between big A, little a, plus, now we do it for B, alpha 1, big B, plus alpha 2, little b, plus the dominance deviation between big B, little b, all the way out until we get to the very, very last thing. So that's what we've done. We've now broken up genetically the contributions of each of those alleles relative to their genotypes, okay? And we've imagined we have done this. We've obviously not actually done this. We're pretending we have done this. And so this is the genotypic effects. Now you'll notice that since they are sums, we can collect our terms. We can bring our terms together. So we can write the sum of the alphas, like so. So from I equals 1 to the number of alphas is equal to alpha sub I, plus now the sum from j equals 1 to n, and these are the delta terms I, j. Alright, so now we have just added, we've collected all of our dominance terms here, we've collected all of our average terms here. Now I've purposely avoided using the term additive, but that is what these represent. As if you've heard about additive genetic effects, that's what these are. Additive genetic effects is just another way of saying the average genetic effects of that allele. So these are our additive effects, and these are our dominance effects. Now let's write this in a fairly simple way that you might have seen before. So G sub I is equal to the additive effects of I plus the dominance effects of I. And now we've done something quite incredible. We have broken up, as I've said many times, we have broken up those two things. We've taken this and we've divorced the genotypic effect from the dominance effects. So let's write this out in a little bit more of a neat way. So keeping our model up here, now we are claiming that the phenotype of individual I is equal to the genotype of individual I plus the environment that individual I is in, bringing that E term back. And then we can break up the G and write that as A sub I plus D sub I plus E sub I. Okay. And that's the model. That's the effect of the genotype, is it's the additive effects plus the dominance deviations plus the environmental effects. Now again, the way that we're using dominance here and the way we're talking about additivity might be a little bit different than how you think about it. But it's actually quite the same. Because if the alleles were exactly additive, then they would fall right along that regression, and the dominance deviation term would be 0, right? But if there is some degree of dominance, and it doesn't have to be absolute, it could be a little bit of dominance, then this term is some value other than 0. Because now there's going to be residual variance that doesn't fall on that 1-to-1. And then of course finally we end up with some environmental effect. So that's our model. That's pretty incredible. So now what we want to do is estimate the phenotypic variance, right? What is the amount of variation within the population? So we can write this as the variance of P sub I is then equal to the variance of A sub I, this is the additive genetic variance, plus the dominance variance, plus the environmental variance. And that's our model. Okay. So now we have a model that predicts the traits and a model that predicts the variation in that trait. All right. So from this, what we now want to do is be able to make a prediction of what offspring traits are going to be, given that we know the value of a relative, of some kind of relative. And in addition, we want to see, can we actually estimate any of these values given that we know the covariance between them? Okay? And that is where the real magic of this model is going to reveal itself. So let's say we want to know the covariance between these two individuals, individual I and individual J. And individual I and J are made up of these components. This is what we're assuming that the individual is constructed of. And so the covariance between I and J is what we're ultimately interested in estimating. What is this covariance? Now, a little trick that I should tell you, a little sort of statistical thing that's going to really, really help us reveal something here, is that the covariance of two values equals the variance of their shared parts. Let me show you what I mean. So let's say we want to know the covariance between x plus z and y plus z. What's the covariance between those? Well, it turns out that it is equal to the variance of the part that they share, which is z. Now keep this in mind, because I'm going to write up here that the correlation, if we remember from all the way back, the correlation under linear least squares is equal to the covariance divided by the variance. So if the covariance is equal to the variance of the shared parts of these two individuals, then what we need to know is what are the parts that are shared between them, right? And that allows us to estimate the variances of those parts. So individual I and individual j, which of these terms do they share together? And how much of those terms do they share? Well, it turns out that the amount of terms that they share with one another is equal to their relatedness. How related are they between one another? So, what's the covariance between these? Well, it's equal to the variance of A that they share, which we will write multiplied by f sub 1, plus the variance of D that they share, and we'll write that as f sub 2. Now for the moment, we're going to ignore the environment. We're going to say that the environment is random. It's something that they don't share. This is like drawing values from a random distribution. Obviously, this term can itself also be shared, and that adds some additional complexity, but for our purposes, and to keep the math as simple as we can, we're going to assume that the environment is independent in them. So these are the two things that they may share between them. How do you share the additive variance? So what does F1 equal to? F1 is equal to what we call relatedness. Remember, the additive variance is the average effect of an allele. Okay? The average effect of an allele is what you get from your parents. Your parents give you one allele. So the relationship, for example, between brothers and sisters is 50%. F1 would equal 0.5 if you are comparing brothers and sisters, because they have a 50% chance of getting the exact same alleles from a parent. That's, I think, relatively straightforward to understand. The second one is a bit more difficult. The second one is called the coefficient of consanguinity. The coefficient of consanguinity really is a fancy term for inbreeding, okay? Because you can think about it like this. Imagine, again, we're comparing brothers and sisters. So what do you have to share the same genotype? That's, in effect, the only way that you are going to have the same dominance deviations is because, remember, the dominance is the measure of the genotypic effects. This is the genotypic effects. These are the allelic effects. So how can you share the same dominance effects if you are brothers and sisters? What that in effect means is that you got the exact same allele as your sibling did from both parents. So let's say your parents both have a big A allele, and you got both of those big A alleles from your parent, and then your sibling got both of those big A alleles in them. In that way, your genotypes are both big A, so you have the same genotypic effects. Now, the probability of that happening is 0.5, that's the probability of getting the first allele, multiplied by 0.5, the probability of getting both alleles. So 0.5 times 0.5 is 0.25 if you are brothers and sisters. Okay? So F2 in this case would be equal to 0.25, right? So again, brothers and sisters would be the variance times F1, which is 50%, plus the variance in the dominance times F2, which is 25%. Okay? And that would give you the covariance. But let's imagine the case in which instead of comparing brothers and sisters, we compare parents and their offspring. Why might we want to compare parents and offspring? Remember, the relatedness is the allelic effects, the average allelic effects. You are 50% related to your parents because they give 50% of their alleles to you. Okay? So, for the parents' case, 50%. However, what is this value? How inbred are you relative to your parent? You cannot have the same genotypic effect as your parent. You can't share the variance in the genotypic effect from your parent because your parent only gives you one allele. They just give you one allele. Because of that, the dominance variance is equal to 0. There is no dominance variance from parents to offspring, because parents give you alleles, they don't give you genotypes. And this is a measure of the genotypic effect. Hopefully, we see how magical that can be. So I'm going to write this out so we can see the algebra. So now imagine we are measuring the covariance between parent and offspring. That is now going to be equal to the variance, the additive variance, multiplied by 1/2, because half of your genes come from your parent, plus the dominance variance multiplied by 0. That means this term falls out of the equation. And what you're left with is the additive variance divided by 2. That is the final thing. Now notice something incredible. We can take this additive variance and we can plug it back into our slope. We can plug it back into the slope because the covariance is here, and that covariance is equal to the additive variance over 2, which is here, right? So we can rewrite our slope as R is equal to the additive genetic variance divided by 2 times, what kind of variance would this be? What is the variance here? That's the phenotypic variance. That's the phenotypic variance in the parent, so that's P, right? And now R is equal to the additive genetic variance divided by the phenotypic variance. What does this term represent? If you've taken any quantitative genetics, or if you've had some like intro genetics classes, what this should immediately jump out at you as is heritability. This is heritability. This is what we mean when we say, is a trait heritable? It's the additive genetic variance divided by the phenotypic variance. So what are the implications for this? Why is this so important? Why does this matter so much? Remember, when we were talking about Pearson's model, right, and we wanted to know how effective selection is going to be, the slope is what determined that. The slope determined how much we could shift the average of a trait, and that slope we measured as R. Well, now we have demonstrated that under Mendelian genetics, R is equal to heritability. And heritability fundamentally is a component of the additive genetic variance. Not the dominance variance, the additive genetic variance. The variance captured by that linear least squares of those genotypes. And so, because of that, what selection is responding to in populations, which is what we showed in Pearson's model, that's the response to selection, it's responding to the additive variance. And that's where the magic finally pays off. So we see that the response to selection that Pearson estimated is equal to the selection differential, which is the difference between what the average being selected and the original average, multiplied by the regression coefficient, which we now know is equal to heritability. And this, ladies and gentlemen, you should recognize because it is called the breeder's equation. This is the breeder's equation. And it is, in effect, just Pearson's model recognizing that that regression coefficient is an estimate of the additive genetic variance. Now, we have come out of this magical world that Fisher has asked us to go down, right? Where we assumed a bunch of things we can never know. We can never know those things. And yet, at the end of the day, we have emerged with something knowable. We have emerged with something that we can actually make precise estimates of. And in fact, with an equation so important and so powerful that farmers and livestock breeders have been using it for almost a century. Right? And they've been using it to predict how their crop or how their livestock is going to respond to selection. And it's why estimating this heritability, which is in effect estimating this additive genetic variance, is so important. Because it tells you how a population is going to respond to selection. And then lastly, one of the sort of key classic ways of estimating that additive genetic variance is just by looking at the correlation between parents and offspring. That the slope of that line is heritability, right? So you just plot those two against one another, and that enables you to get a measure of the additive genetic variance. So we can estimate genetic effects, actually we're estimating allelic effects, and we have not sequenced a single individual. We've not looked at a single gene. We don't know what any of the genes are doing, right? We don't know how many of them there are. In fact, we're assuming that there's an infinite number of them, and each of which are contributing extremely small effects. And yet, by doing a little bit of mathematical magic, we've walked out on the other side and demonstrated that Galtonian statistics and the Mendelian genetics are actually completely compatible and explain one another. And that ultimately, what matters in evolution, what matters to selection, is the additive genetic variance. That's the power of Fisher's 1918 model, and it's the power of Galtonian statistics. So, with that long kind of walkthrough, let's now return to the desk. I'm tired of standing. I've been standing for so long. We're going to return to the desk, and we're going to finish up by talking about some of the historical developments after Fisher's work that finally began to bring quantitative genetics back into the fold of evolution. We're going to introduce you, again, to some more figures that you've probably never heard of, but hopefully, at the end of the day, will be beneficial for us to answer this final and lasting question of why bother finding the genes? So in the decades following Fisher's 1918 paper, a lot of individuals expanded on it and elaborated on it and tried to work out all the little details of it. And sure enough, eventually it became known as the infinitesimal model. Now, Fisher didn't call it the infinitesimal model. This is something that was applied to it by later people. But I think it's important for us to review the sort of major implications of the infinitesimal model because it's going to be really relevant to us here shortly. So firstly, as I said at my little whiteboard scene, the infinitesimal model assumes in effect an infinite number of genes are impacting a trait, and because of that, they are each doing so with infinitely small effects. Infinite is like with the mathematical assumption, but really and truly it works just as well if there's many, many, many genes, is the way you can kind of practically think about it, is lots of genes are impacting that trait. And so as you increase the number of genes contributing to the trait, their individual effects must also get proportionately smaller, right? Another key implication of this model is that the present standing amount of phenotypic variance that you see is actually much smaller than the amount of variance possible within that population. The amount of actual genetic variance far exceeds the amount that you actually see. And the reason for that is because if there are infinitely number of genes impacting a trait with, any number of allele at each one of those, then the number of possible combinations of those alleles exceeds the population size, right? There are far more ways to arrange those alleles than there are individuals capable of expressing those effects. What the implication of this is that as you shift the population, let's say if you select individuals from one average, you can get a continued response to selection, and basically an endless response to selection under the infinitesimal model. And we see that in agricultural settings. We've seen a consistent shift in the average by 10, 15 standard deviations from the original mean because there is so much variance that exists sort of unseen because there are so many genes contributing that, again, are far outnumber the number of individuals in the population. This also means something really radical, and that is that if you think about in sort of the classical Mendelian sense, in the classical population genetic sense, we often model selection acting on individual alleles. So individual alleles have a selection coefficient, and that's basically their fitness effect. In the infinitesimal model, we're assuming that the effects of each allele are basically infinitely small. If that's the case, then selection on each individual allele must also be infinitely small. It must have a very, very tiny effect. What that means is that the dynamics that are actually going to be shifting individual allele frequencies is not selection, it's drift. Again, at the individual level, selection is too weak, so genetic drift should be dominating individual allele frequencies. This has a pretty interesting implication, right? In that despite the fact that selection is acting on the phenotype, the underlying allele frequencies actually fluctuate mostly by genetic drift. And I'm gonna show you some implications of that here shortly. So there's a kind of effective neutrality at the genetic level, even though selection is acting at the phenotypic level. This also means something really, really cool, and that is that the expected amount of genetic variance within a population should be dictated by the effective population size. That is to say that selection should have a very, very weak effect on changing the amount of genetic variation within the population. And the main thing that is going to be reducing the genetic variance in the population is actually inbreeding, or genetic drift. And so, that means that the standing amount of genetic variance should be proportional to the population size. It also implies that over time, the amount of genetic variance should stay relatively constant, because you're going to, in effect, reach a mutation drift equilibrium even at the quantitative level. Because again, what's dictating the amount of genetic variance is drift, so if you have a constant input of mutation, a constant loss of variation due to drift, you should reach a sort of mutation drift equilibrium. This also has big implications for the molecular clock. It means that the rate of substitution should roughly be proportional to the mutation rate with a slight bias. And this is probably why the molecular clock works decently well, and why the rate of substitution that we see across organisms largely appears neutral. This is true despite the fact that we're assuming, in the infinitesimal case, a model in which selection is shaping the phenotype. It's just that drift is shaping the genotype. And again, that feels like a kind of interesting paradoxical thing to say, that those two things can be true, and yet that's exactly what the infinitesimal model predicts. Okay, so that's Fisher's infinitesimal model, all the implications of it, the math of it. I've spent way too much time on this. This video is going to be way too long. This is what I get for trying to do things unscripted and including a whiteboard section. If I include a whiteboard section, it's going to be way too long. I take too long at the whiteboard. Now let's move on to what happened after Fisher 1918. So the rest of the Mendelians basically ignored it. They didn't do anything with it. The Mendelians went on doing their Mendelian genetics, doing their crosses in labs, working out the structure and functions of genes, right? Like, that was the avenue that they took. But there was a really important character that sort of emerged shortly after Fisher. In fact, I think he got his PhD in 1922, and this is J. Lush. So J. Lush was an animal geneticist, and he had a big interest in breeding, right, in livestock breeding. And he wrote an extremely important book in 1943 called Animal Breeding Plans. He basically went around the country and advocated using quantitative genetics to better improve livestock. Because what most of livestock breeding had been before quantitative genetics had been just kind of up to the whims of the breeder. The breeder's like, oh, this looks pretty good, so I'll just breed individuals that have this. But there was nothing quantitative about it. They couldn't actually make predictions about the rate at which things should change. They could easily be befuddled by the fact that they would have lots of individuals that didn't seem to have the trait, right? There was this, it was very vibey, right, how livestock breeding worked. And it worked, it was just very inefficient. And so J. Lush was like, look, instead of going on the whims of the breeder, we can do this quantitatively. We can actually apply quantitative genetics to make accurate predictions about what he called breeding values. And breeding values are basically what we meant when we said the average genetic effect. And that's going to tell you something about the response to selection. And some like really powerful examples of this is like being able to estimate the breeding value of a bull for milk yield. That's crazy because bulls don't give milk, and yet you know they hold the genes to give milk. And so if you know their average genetic effect, you know their breeding value, then you can breed bulls that are going to sire cows that will give lots of milk, right? And that's the kind of thing that J. Lush's approach was able to do. I want to show you a couple of graphs about just how successful quantitative genetics has been. So this is a bunch of different staple crops. On the y-axis is the amount of yield per ton by hectare. And you can see across all of these staple crops, whether it's wheat, whether it's oat, whether it's barley, whatever it might be, we've seen this steady increase in the amount of yield. This is only going back to the 1960s. So just a consistent increase, sometimes tripling. So look at wheat, for example, goes from 1 ton per hectare in the 1960s all the way up to 3 tons per hectare in the 2020s, right? So that's one bit of powerful evidence. So here's another one, and this one is like really wild to me. So this is from chickens, and the graphs all here start in 1925, and then they go up again to the 2020s. And I just want to like try to situate this in human terms to try to show you just how wild these graphs are. So the first one is the day to market age. So when does the chicken reach the size that you can take it to market? In 1925, it took over 100 days before that chicken reached the size that you could take it to market. And it was like almost 120 days. In 2020, you could take a chicken to market in less than a month. Under 30 days. That's a wild increase in the rate at which they reach maturity. That's akin to a human being like an adult at 4 years old. Imagine a 4-year-old adult running around. Now, what's incredible about this is not only do they reach maturity much, much faster, but they get so much larger when they reach maturity. So again, here it is on the x-axis. You can see a 3-fold increase in the average size of a chicken when it reaches market age from 1925 to today. It would be like a 4-year-old that is an adult and is like over 400 pounds. And not only that, that 4-year-old adult over 400 pounds is like pure muscle mass. Like it's not even like they're fat or something. It's like muscle that they have built up in just 4 years. That's crazy. Not only that, but check out this graph. This is the amount of food relative to the grams of meat that you get back. So you can think about this as the payoff. So how much food do you have to feed this chicken relative to the amount of meat that you're going to get back from it? Notice that by 2020, you almost get the same amount of meat back as food you are feeding the chicken. There's an almost 1-to-1 payoff. So for every bit of grain you give that chicken, you get a gram of meat back. That's crazy efficient. Crazy efficient. To me, this was like one of the most startling bits of data that I've ever seen. And in case you think that this means that chickens are like dying en masse and that they're like suddenly so much worse than they were and like barely any of them survived to market, here's the mortality rate in 1925 compared to the mortality rate today. That is a precipitous decline in the mortality rate, all the way down to under 5%. So not only do they survive better to market, they get there faster in less than a month, and they get there 3 times larger than what they did in 1925. This is the power of quantitative genetics. Now, I'm not saying obviously anything about the sort of morality of chicken breeding. God forbid I do anything like that. That's a wholly different question. I'm simply saying that applying the principles of quantitative genetics has enabled us to breed chickens like never before, right? Like that rate of increase is nothing like anything we've ever seen from just farmers being like, oh, I think this is a good-looking chicken, I'll breed this one. Applying this quantitative approach just had a dramatic change in our ability to raise crops and livestock. So I hope that sort of practical diversion helps you see just how useful the infinitesimal model is, despite the fact that its basic premise is that there's an infinite number of genes, which we know that's not real. It assumed that we could do a bunch of regressions on individual genotypes. You know, you can't do any of that, right? It assumed all this sort of magical thinking, but we came out of it on the other side with something extremely practical, perhaps the most practical thing that we've ever been able to come up with. J. Lush has this really incredible quote that I think is quite relevant here from Animal Breeding Plans, in which he makes this argument that even if the animal breeder knew every single gene and what every single gene was doing, it wouldn't change what the animal breeder would do. The animal breeder is still just going to estimate the heritability of a trait. And they're going to do it exactly in the way that Fisher did it. You're going to do it by either some kind of regression method, you're going to do it by some kind of linear model, you're going to estimate heritability. And in fact, I should point out that the sort of regression technique for estimating heritability is a little outdated at this point. Nowadays, most people use a more general linear model. This is called the animal model. Maybe I'll make a video one day going through sort of the details and the sort of nuts and bolts of the animal model. Really, really cool stuff. That's what most people are going to be using nowadays. But underlying the animal model is still the basic assumption of Fisher's infinitesimal. So that's the sort of the practical history behind the infinitesimal model, and why animal breeders basically didn't need to know what the genes were. They didn't need to look for genes. Nowadays, you do have things called genomic prediction, but what genomic prediction does is it's not about finding genes, it's actually just about estimating relatedness. So basically, you can improve your prediction of breeding values of average genetic effects if you know what alleles were inherited between parents and offspring. Again, you don't know what those alleles are doing, and you don't actually even care because what they're doing is so small that it doesn't even matter, right? You're just using it as a way to improve your prediction of their breeding value. Because if you know exactly what alleles were inherited, and you know what alleles the parents had, then you get a stronger prediction than just guessing, well, 50% of the time they're gonna have the same alleles, right? You're improving that sort of prediction there. And that's actually what the animal model uses, is it uses that kind of sequencing predictive value. So again, we do the sequencing, but it's not at all to figure out what the genes are doing. It's just to estimate breeding values, right? Which again, is a pretty wild thing that we do. So enough about sort of selective breeding and that kind of practical history. Hopefully you're convinced of that. If you're not convinced that this model is powerful, and in fact probably the most powerful model in all of biology, then I don't know, live on just like scavenging nuts and hunting deer for the rest of your life and see how much easier it is to live that way. So what I want to talk about now is, as sort of the timeline is moving forward and we're coming out of the 40s and 50s where the sort of practical animal breeding has really taken off and has really become this powerful thing, it has developed, as I've tried to argue throughout this video, it has developed largely outside of the public eye. People aren't seeing this happening because what's obvious and relevant and at the forefront in sort of public education and public discourse is Mendelian genetics, right? It's the discovery of DNA is happening in the 1950s, right? The structure of DNA is uncovered. Gene regulation is being uncovered into the 1960s. Nobel Prizes are being handed out in mass for working out the genetic code. People like Sidney Brenner, people like Jacques Monod, right? Like all of these characters are rising to the forefront at this time. And so while that's happening, right, quantitative genetics is basically being developed sort of under the radar of all of this. And there's this figure that emerges in England in the 1960s, and his name is Alan Robertson. And Alan Robertson, I think, finally brings quantitative genetics back to sort of population Mendelian genetics, the sort of the classical allele by allele approach of the old population geneticist approach. So Robertson was interested in what is the limit to selection. We've been able to continuously select for traits. We've increased the mean by 15 standard deviations in tons and tons of crops and livestock, right? What's the limit? Where do you reach the sort of wall and you can't progress any further? Robertson approached this in two ways. First, he calculated, well, what is the response to selection in one generation? How much do we expect it to respond? And this is basically the breeder's equation. But he showed it as delta z bar, this is the change in the mean, as equal to the additive genetic variance, V sub A, multiplied by beta, which is the selective gradient, okay? So that's the response to selection in one generation. Now, under the infinitesimal model, right, assuming the sort of classic quantitative genetic model, what is the limit to selection? Well, it turns out that the limit to selection is the limit to the amount of genetic variance in the population. And that limit is set by the effective population size. And so, what Robertson found, quite incredibly, is that the net change in a trait over time that's being selected is V sub A, the additive genetic variance, multiplied by the selective gradient, beta, multiplied by 2 n sub e. So the response to selection is bounded by the effective population size. For a really, really small population, that means there's not a lot of genetic variance, and you can't change the mean that far. For an extremely large population, in populations in nature, for example, the effective population size may be is in the thousands, if not hundreds of thousands. And that means that the limit to selection is pretty far off. Right? You can push that mean really, really far if you've got a very large population. Now, this has really important implications for how we understand what selection is doing at the allelic level. So remember that I said that selection at the allelic level is effectively neutral. And so drift is dominating the dynamics. And then we're seeing at the sort of phenotypic level that drift is also sort of setting the bounds. The effective population size is setting the bounds of how far you can move the average of that trait. And so what does that tell us about selection at the allelic level? Well, what Robertson found is that selection is acting at the allelic level, but it's extremely slight. And in fact, the degree at which it's acting is 2 multiplied by the effective population size, N sub E, multiplied by the selection coefficient on each and every individual allele. Now that's an extremely small effect, right? But what that does is it adds a slight bias in the probability of fixation of any given allele. And so if you were to compare 2 different distributions, one here that is just a neutral distribution, those alleles have a selective effect of 0, and then over here is one in which there is some selection. It's extremely tiny, but there's some selection acting here. And again, drift is dominating the dynamics, but if you squint, you can see that the distribution actually is slightly biased. It's not actually giving you what's expected under neutrality. And that's what Robertson showed, is that selection is acting on the individual allelic level by just slightly biasing their probabilities of fixation. So that's Robertson's famous 1960 paper, which first finally started integrating back in a population genetic approach. Into the 1970s, you start getting the work by people like Russell Landy and Steve Arnold, and what they do is they bring in natural history. They start investigating how quantitative genetics can be applied to organisms in nature. They are estimating heritability in things like garter snakes and fish and Darwin's finches, right? Like those sorts of organisms in nature instead of the farm. And what they're finding is that quantitative genetics also seems to work in that setting as well, that there's a large heritability for many traits in nature. They find something really fascinating, and that is that the heritability of traits tightly associated with fitness, so things like life history traits, tend to be very low, tend to have low heritability, where traits that are more loosely connected to fitness have high heritability. Now, this is exactly what we would expect if selection is mostly acting on additive genetic variants for traits that are associated to fitness. Because in effect, what selection should do is it should slowly sort of weed out any of the additive genetic variants associated with fitness. And that's exactly what we find. And this is what Fisher's fundamental theorem predicts as well. I've done a whole video on that, link in the description. So that's to sort of push us forward, because this video is already getting too long. I want to take us now back to that seminar that Nick Barton gave and see if we can understand what in the world he was talking about. So one thing that's kind of incredible about the infinitesimal model is he, as well as Alison Etheridge, have done sort of an in-depth dive to the guts of the infinitesimal model and what it assumes. And one of the things that they were really interested in figuring out, when selection is most efficient, if we're thinking about an infinitesimal regime. So, for example, is selection really inefficient in an infinitesimal regime because, at the individual allelic level, it's barely detectable? Or is selection more efficient when it's like one locus with a really big effect, right? Like when is selection the most efficient? So one of the ways that they tried to approach this is try to come up with an objective way of estimating selection's efficiency, right? How well selection can drive adaptation. And they did this by introducing a measure of information, of genetic information. Now, I know from like creationist circles, the idea of information in the genome is a whole can of worms. But they did it in an objective way. And their measure is actually closely related to Shannon's information index. And it was first used by Kimura, actually, in 1954, I think. Kimura was the first one to introduce this. But this is the way you can think about their measure of information. So information is, in effect, a bias in the distribution of allele frequencies relative to what you expect under neutrality. So actually, it's just equal to the log of the ratio between the observed distribution divided by the distribution expected under neutrality. So for example, this is a distribution of allele frequencies expected under neutrality. You can simulate that through time, and you can see how it should look based on no selection at all acting. And then this is a distribution when selection is acting. And you can see that it is biased away from neutrality. So in effect, what selection has done is it has increased the genetic information. There's more information in this system because it's less and less and less like what you would expect under just a random system like with genetic drift. And so what Barton did is he said, okay, when is selection best? When can selection maximize the amount of information within a genome? Now, the trick here, the key to understanding this argument is that there's also a cost to selection. There's a cost to trying to maximize information. And the cost is the reproductive success of individuals. Because for a population to sort of sustain itself, selection can't be too strong. And this goes all the way back to Haldane's famous 1957 cost of selection. Because an individual can only have so many offspring. So if selection is really, really strong, and it's just like purging everyone, right, then the cost gets really high. And so you can get a lot of information, but you're getting that information by paying a really, really high cost, and not all populations can pay that cost. And so the cost of selection is proportional to the number of individuals that are basically dying, right? And you can think about it at an allelic level as how strong selection is acting at each individual locus. It's a kind of substitution load. And so when you think about the metric like that, you've got the cost of selection, and then you've got your measure of information. And those two things together is how you can measure the efficiency of selection. Maximize the amount of information while minimizing the cost. Where does that occur? Here's some graphs. On the y-axis here is the amount of information given the cost. And then on the x-axis here is the selective intensity. All right, so how strong is selection acting? What you can clearly see in this graph is that selection is most efficient. It can maximize information within genomes best when it is extremely weak at each individual locus. Because what that implies, it's not incurring a very large cost, right? Because it's not harming you because it's so weak at the individual locus level, so the cost is very, very low. So when you consider those two things together, selection is actually the most efficient, not in the one locus big effect situation, but in the infinitesimal, where each individual gene is contributing so little to that trait that it's virtually undetectable. And that's really Barton's big take-home message here. It turns out that selection is the most efficient when we can least detect its action. And that kind of sucks, because it means that seeing natural selection acting is going to be so much harder than just looking at the genome. When you look at the genome, you won't actually see rampant signatures of selection. Even if selection has been rampant, even if it has been driving all of these alleles, it has been driving phenotypic change, you won't see it at the genomic level because function is so diffuse, it's so spread out. Okay, so where does that leave us? We have now talked about these two distinct branches of genetics. We talked about Mendelian genetics, we've talked about the infinitesimal model and quantitative genetics, and how they've basically progressed independently for a very, very long time. Even though both of them have made extremely massive impacts on science. Only now, only today, are they starting to creep back together. And that brings us back to those few papers that I kind of opened this with. Those papers were basically arguing that the classic Mendelian approach of trying to find the gene leading to the phenotype has been increasingly unsuccessful for complex traits because it looks like there's infinitely number of genes impacting it. To go to Pritchard's terminology, what it looks like is the omnigenic model, the all genes model, seems to be the one that best explains complex traits. And that if you want to account for the heritability of complex traits, you basically have to use all the genes to get there. And if that's the case, then suddenly the individual gene doesn't matter anymore. It's doing so little that it doesn't actually benefit you to know what the molecular basis of that gene is, of like what it's actually doing, how it's regulating the network doesn't seem to really matter. The argument, the statistical heaven argument that Nick Barton is making, is that instead of trying to find the molecular basis, instead we just need to know what is the heritability of these traits. And if we can maximize our ability to associate breeding values with these individual traits, so what is Alzheimer's risk? What is diabetes 2 risk? That if we can find out the average genetic effects across individuals, then that will enable us to make predictions about risk factors. Right? We can make a prediction about what is your chance of getting Alzheimer's, given the breeding value that you have. But what might be forever out of our reach are things like gene therapies, right? These sort of targeted genetic therapies that are going to go in, find like the molecular basis or the chemical basis of each one of these alleles, and we're going to stop up the proton pump in this cell because we know that it's powering this cascade effect. Like all of that stuff is... It's not going to happen. And it's not going to happen because at the individual genetic basis, the effect is too tiny to make a difference. So the whole question about, do genes matter? Obviously is a little hyperbole, but only a little bit. Little by little, what quantitative geneticists are trying to do, and what these papers are sort of arguing for, is getting molecular biologists on board with the idea that statistics can tell us something powerful about inheritance and about evolution. So, does the gene matter in evolution? What do you think? Tell me your thoughts on quantitative genetics and my sort of off-the-cuff way of delivering this video. Hopefully it was informative. I just could not bring myself to sit down and write a script. I've been far too busy, but I had all of these thoughts in my head and I wanted to get them out. And so I thought, you know what? I'll just sit down and I'll just ramble at the camera until I get everything out. And that is exactly now what you have seen. So it's probably disconnected and wild and crazy, so ask questions in the comments if you are confused. Also vote for, I don't know, your favorite place of seeing me sit and your favorite t-shirt that I was wearing. Thank you all so much for being here. What's coming up on this channel soon is I am going to be writing a script for the 50th anniversary of The Selfish Gene. I have a lot of thoughts about this, and I'm pretty excited for that video. This should be coming out hopefully relatively soon. But yeah, thank you all so much for being here, and I will catch you next time.